Good morning and good afternoon, ladies and gentlemen, and welcome to the webinar on real-world research on medicinal products, key contribution to the European Network of Centers in Pharmacoepidemiology and Pharmacovigilance. My name is Kelly Plushke, and I am pleased to facilitate the meeting today together with my colleagues Valerie Muldoon, Juliana Vogt, Audiovisual and IT. Before we start, I just would like to say a few words on housekeeping notes. May I kindly remind all participants to mute their audio when they're not speaking, as this will help the smooth running of the meeting and will minimize background noises. The webinar is being broadcast live on the EMA website and is being recorded as well. And we will publish at the end of the meeting the recording as well as the speakers' presentations. Should you wish to ask any question, you can use the chat box as well as you can raise your hand at the end of the presentations or during the Q&A sessions. You may turn on your cameras. There is no obligations, but it's always nicer to see the faces of the speakers. So I leave it to you to decide. Having said all that, I wish you a very good meeting, and I will hand over to the two co-chairs of our session today, Jean-Mario Candore and Susana Perez-Gotan. Thank you very much, and have a good meeting. Jean-Mario and Susana, over to you.
Thank you for introducing the webinar. My name is Gianmario Candore, and I'm a statistician working at the European Medicine Agency, and I will co-chair this meeting together with Susana. Susana, are you online?
Yes, good morning, everyone. My name is Susana Perez-Gotan. I work at RTI Health Solutions in Barcelona, and it's my pleasure to co-chair this meeting with Gianmario.
Great, thank you. So to introduce this meeting, just a few words on what are the objectives. As you know, EMA is further developing the interaction between the EU regulatory network and the academia. This both to provide to identify some needs where where academia can help to satisfy regulatory challenge. And this webinar has four main objectives. And if we go to the next slides, we can show the objective. The first objective is indeed to raise the awareness of Ncep through academia and the Learned Society. The second objective is to highlight the Ncep contribution to pharmacoepidemiological research, and then to learn from the member experience of Ncep and understand how the network can support academia and Learned Society. And finally, we will have time to discuss a methodological challenge for observational studies. So in the next slide, we can see the agenda for today. It's a quite rich agenda. We have 8 presentations, 2 hours available. If we keep time, we will have half an hour at the end for a question, but we also foresee that we will have 1 question at the end of each presentation. So without losing any other time, I would like to introduce the first speaker, that is Xavier Kurtz. He's the previous co-chair of the Ncep steering group, and he will talk about what is Ncep and why it's relevant for academia. Thank you, Xavier.
Good morning, everybody. So I'm Xavier Kurtz. I'm working at the European Medicine Agency, and I am a medical doctor and epidemiologist. So I will first introduce what is Ncep and why it is relevant to academia. Next slide, please. So Ncep is a network of centers which are active in the field of pharmacoepidemiology and the field of pharmacovigilance in Europe. And the main activities of Ncep and function of Ncep is to bring together expertise and resources in pharmacoepidemiology and pharmacovigilance, and then to support collaborations between the different centers, different experts, and different activities. It also facilitates the conduct of high-quality, multi-center, independent post-authorization studies with a focus on observational research, and there will be throughout this session examples and explaining how this high-quality research is being support it. And then it develops and maintains methodological standards and governance principles for research in pharmacovigilance and pharmacoepidemiology. It should be clear that Ncep does not assess specific safety or effectiveness issues of authorized medicinal products, so it does not replace regulatory authorities. But of course, it discusses and it develops methods for studies, for doing the studies, although it does not take decisions in terms of safety or effectiveness of products. Next slide. So currently there are 198 centers which are public organizations, university, hospital, government, or charities, and or CROs or consultants. So the Ncep centers do not include private companies, so it does not include, for example, pharmaceutical industry. They are not included as members of Ncep. And you see the map that centers are spread out all Europe, all over Europe. This is mainly Western Europe, but Eastern European countries are now increasingly involved. There are also 31 networks, so networks of research network or networks of organizations, and 153 data sources, and the network is steadily growing. Next slide. This is reflected by the growing frequentation of the Ncep website, so ncep.eu, which is receiving a larger number of visitors per every year. So this is increasing, and there is information that is on the Ncep website that is being used and accessed by publicly and also by all the members. Next slide. So why should you register as an Ncep center if this is not yet the case? By being an Ncep member, you will be part of an active community of scientists, and this community discuss methodological and operational issues. And what do the centers told us is that these issues are issues that are being discussed and in Ncep, but not discussed elsewhere. So this is a very active, dynamic group of people creating within working groups and special interest groups that are discussing topics in pharmacoepidemiology and pharmacovigilance. You will be also directly informed of research projects or potential funding opportunities or calls for collaboration and EMA announcements. So the Ncep Secretariat sends immediately by email to all Ncep members announcements that, for example, there will be next week, there will be a new announcement for framework collaborators, for contractors for EMA-funded studies that will be sent to all Ncep members. This will also contribute to the development and dissemination of the best methodological standards. So it is important to contribute to this development of the best methods, and also to contribute to develop good governance of pharmacoepidemiological studies in Europe, for example, by agreeing and by discussing and by putting forward principles of independence of academic investigators from the influence of study funders, such as pharmaceutical companies, publication rules, or control agreements. These are topics that are important for the success and for the quality of pharmacoepidemiological studies in Europe. And then as Ncep Center, Ncep members, you will be involved in the discussions and the creation of these principles. What does it mean to be an Ncep partner? First of all Ncep partners are registered in the Ncep resource database, which is a public database. There is also a commitment, and being a Ncep partner means a commitment to adhere to the principles of the Ncep Code of Conduct and Ncep Guide on Methodological Standards in Pharmacoepidemiology. So this means a commitment to apply best practice in the conduct of pharmacoepidemiology. There is also a commitment to register the post-authorization studies in the EU PASS register for transparency, and to participate in the development of research and good practice standards by contributing to or commenting on draft proposal prepared by working groups or the Ncep Secretariat. And also there's a commitment to collaborate with other Ncep partners to be part of the European community of pharmacoepidemiologists and experts in pharmacovigilance. So one of the most recent publication that has been made, and it is really the a sign of the collaboration, an example of the collaboration that has been taken place within Ncep, is a publication on different strategies to execute multi-database studies for medicine surveillance in real-world setting. And this publication is now used as a reference on the different models for accessing database that may be presented, may be available in different formats. So there are different models that can be applied and different principles and concepts and methods that can be applied. Just the last slide is on the organization. So this is, I'm not going to be to go into the details, but so there is a steering group, which is the main decision-making body of Ncep, and the steering group is composed of 17 members, including 6 that are elected by the centers and also 8 but appointed by different committees, but also different learned societies like ISPE, ISOP, ISPO, and there are also representatives from the FDA, from Health Canada as observers. And so there are a number of working groups, task force, special interest groups, and at least once a year we organize a plenary meeting with all research centers. So I will then pass the floor now to Francesco Salvo, who is one of the members of the Ncep steering group, elected by the Ncep centers, and we will discuss how does Ncep support academia. Francesco.
Thank you very much, Xavier. I'm Francesco Salvo. I'm from the University of Bordeaux. I'm associate professor of pharmacology, and I'm working in pharmacoepidemiology and pharmacovigilance. So how Ncep could support the academia. First of all, Ncep provide principles and tools, in particular by promoting transparency for the registration of the study, but in particular the publication of the protocol and the results of the study that could increase transparency and reduce as possible post hoc analysis. With the code of conduct, we help in clarify roles and responsibility of all parties involved in for public health benefits. So we assure independence of the studies, and of course, and this is the most important advance in the last years, we provide standards to increase methodological quality of the study and of the protocols, in particular by providing a checklist for study protocols. Next slides, please. So this is just an example. It's really a little of the guide of methodological standards in pharmacoepidemiology, in particular here for providing some key aspects for research for the COVID pandemic. For example, if you look at the point 4, you can see how we can support primary data collection, secondary data analysis, and made, for example, high quality spontaneous report studies. Next slide, please. So this is how this is accepted, I would say, but by community. So we have a single web resource for methodological standards in pharmacoepidemiology in English. In 2020, we have the 8th revision of these standards, and we provide links, for example, to selected articles or topics that we consider as the best methodological guidance. For academia, it is important to have these tools because it could be used for training worldwide. So here you can have a little figure with the use of these standards. We have in 2020 more than 100,000 views and more than 20 downloads of this guidance. It's a huge works that involved a lot of people, 33 authors, for a total of more than 550 reference in the revision 8. So it's really a huge work to provide standards in any kind of observational study design. Please, next slide. I also speak about the register, so the public register that allows the publication of study protocols, first results, or other study documents. So is a huge effort for transparency of study conduction and of quality of result reporting. Next slide, please. This is how it works. Also, our registry. So it was created in 2011, and with a global increase in the first years to arrive to around 250 studies that are collected in this register, with a little boost in 2020, and we passed the line of 300 study registered. Next slide, please. This is the checklist. One of the most important part that is very useful for academia, in particular for doctoral, postdoctoral, and postdoctoral students, to support the best practice and allow, for example, to students or to researchers to have the nice question to design an observational study. Is of course a tool that could help in promoting transparency on studied measures, and of course could be used also by, for example, pharmaceutical companies when submitting protocols to medicine regulators. So it's a global tool that could help people to promote a huge quality of study protocols. Next slide, please. This is a nice paper from Rosa and other colleagues that concerning the code of conducts. So that is very useful to summarize the best practices for scientific independence and transparency in non-interventional post-authorization studies that resume our code of conduct for this kind of study. Next slide, please. It's of course also important to be for regulatory science because Ncep members provide data and occasionally also publication to European Medical Agency that could support drug safety reviews. People from Nsap could also participate to EMA-funded study, to public announcements, and also involved in European Commission FP7 drug safety program. We also promote EMI public-private partnership, and of course with our network we can be of help for ad hoc collaboration for health public interest studies. Next slide, please. So what's next? Ncep is a network that aims to increase and to support collaboration to improve the capacity for conducting multi-center studies in pharmacoepidemiology. We work on innovative methods to access and analyze data from online database, for example, using common data models and also artificial intelligence to analyze this data. In a context of huge innovation in these fields, it is important to interact with new networks. For example, Darwin EU community, or the big data stakeholders, or coordinated registry networks. So we aim to ensure the network to remain focused on the most relevant public health issue and decision taken by regulators, health technology assessment, and other bodies. We aim to include new experts and new centers to embrace relevant new areas of activity, as for example, use of patient registries, social media information, and big data. And the last important part of this speech is also to reinforce collaboration also with non-European researchers and other health decision makers. Thank you very much for your attention. I'm ready for questions if there are some.
Many thanks, Francesco and Xavier. That was a perfect introduction to today. We will have half an hour at the end for questions, but if there is any burning question now, we are happy to take it. No questions, so we can move to the next presentation, where Gianluca, professor of pharmacology at the University of Verona, will go more in detail in one of the tools that was introduced in the previous presentation. So he will show why it's important the EU register of characterization and what are the lessons learned during these first 10 years of use. Thank you, Gianluca.
Thank you, Gian Mario. I'll be presenting in this talk the work that has been taken by the Working Group 3. It's a very active working group exploring issues concerning the EU Pass register and multiple database studies specifically. This is my disclosure of interest, nothing related to the talk I'll be giving. Well, coming back to one of the previous slides, I'll be describing a little bit more into detail the value of the EU Pass register, that is the European Union Electronic Register of Post-Authorization Studies. This is a publicly available register that is everybody can register non-interventional studies. And the focus is on observational research, but we see that actually there are also some clinical trials registered in it. With the aim to increase transparency, reduce publication bias, promote exchange of information. This was particularly important in the period of first wave of COVID-19 pandemic, because a very large number of observational studies have been published, and most of them have been registered in the EU Pass. I think your colleagues later on will discuss a little bit more in depth this item. And also ensure compliance with the EU pharmacovigilance legislation requirements. In fact, all the pass that are initiated, managed, financed by a market organization holder, required by a risk management plan, has to be registered into the EU Pass register, again to promote transparency and to guarantee scientific and quality standards. The EU Pass register has been investigated in a number of publications that basically descriptive publications, but providing interesting information on what are the main characteristics of especially the regulatory mandated observational studies that are registered in the EU Pass register. It's important to explore continuously the EU Pass register, because this can really provide interesting information for the scientific community, for also regulatory bodies. The Working Group 3 that I have the pleasure to lead performed an exercise in the last year and a half to make the information registered in the EU Pass register analyzable and to derive key information from this analysis. It was a really collaborative effort with 14 centers a huge number of quality check and several analysis, also validation of the data collection from different partners. So we had to develop a specific electronic case report form in order to transform some of the information into analytical variable. What's clear is that it's not easy to extract and analyze information from the EUPASS register for some specific type of variables. And you can see in this slide that for some of those variables, the agreement was not really optimal among different validators. So this is also, there is probably also space for improvement in the way the information are registered in the EUPASS register. This is a picture of the studies registered into the EUPASS register till the end of 2018. As mentioned, there is a lot of work in analyzing individually all the studies. So that's why we have been working on studies until end of 2018. Of course, once all the methods we developed for analyzing the EUPASS register are in place, continuous update of the EUPASS register studies can be much faster and easier. Overall, during this period, from inception to December 2019, almost 1,500 studies registered. Most of them, of course, are observational studies. 90% of them are observational studies. Is already an important piece of information here that around 1/3 of the observational studies are based still on primary data collection. That requires, of course, lots of effort and time to conduct. Having a look at different types of studies, here you can see the trend of different characteristics of studies registered in the EUPASS register. The overall the picture is in A, with a cumulative distribution over time of those studies. In panel B, there is a stratification by type of studies, with observational studies that clearly represent most of those studies over time, even more. And panel C, there is the cumulative frequency of observational studies stratified based on data collection, where, as I mentioned earlier, we can appreciate that more than 1/3 of those studies are still based on primary data collection. And also an interesting point is that the number of multiple database studies has been increasing over time, especially in the last period. Some key information that have been derived from descriptive analysis of those studies. A lot of studies are requested by regulators, but there is also a significant amount of studies that are basically registered on voluntary basis by researchers, especially if those studies are funded by a pharmaceutical company. And you can see here for observational studies, this is the case in more than 80% of the studies. And looking at different data sources used, it's really heterogeneous, with electronic health record, claims databases, registries, or more than one of those data sources used in the studies. Another important piece of information that more than half of those observational studies explore risk associated to some medicines, but most of the time these are descriptive studies rather than analytical ones, with just incidence rate of certain adverse events of interest during exposure to studied drug. And good point is that children are also often investigated, 1/3 of the cases, and also other specific population like pregnant women or breastfeeding women with a much lower proportion. But still, important information from these specific subcategories can be derived. Another important point for discussion is the availability of protocol. This is probably a weak point at the moment. It was highlighted in our analysis because more than a half of those studies, and these are the only observational studies, have no protocol deposited. This is a limitation because does not facilitate the exchange of information and also transparency. So this is a point to be highlighted, and the ones registering studies should be invited to register, to deposit also the protocol. Also, orphan drugs are mainly explored using existing registry, but in part also using claims or electronic health records. Another source of information for those studies are publication, and in around 30% of the cases are available. A little bit more in-depth analysis on multiple database studies that has been increasing over time. Well, they are more likely to have a protocol deposited, to receive also the Ncep seal, to be requested by regulator, especially EU RMP1, that are studies imposed as condition of marketing authorization. And also another interesting point, multiple database studies are more likely to be conducted when the study population concern children. Again, this is just for descriptive purpose, but some interesting information can definitely be derived. There are other more in-depth analysis on multiple database studies, and in particular the regulatory outcome of those studies based also on the models that have been adopted for the conduct of multiple database studies. This is based also on the previous publication that was shown by Francesco in the previous presentation. Focus on regulatory outcomes. There is a dedicated task force that is developing methods to evaluate regulatory outcomes of those studies, and a focus also on pediatric population and pregnant and lactating women. As mentioned earlier, there are it's a low proportion of studies, but still it's very informative to analyze specifically those subcategories. And also another task force is on the is exploring the heterogeneity of studies registered in the EU PAS based on the different European countries participating. Just to conclude, well, assessing the EU PAS register requires multidisciplinary and advanced expertise. Maybe there is a space for improvement in data registration in a way the ones that want to register maybe more effectively supported in the accurate data registration, and also should be invited to deposit systematically the protocol, not only if the study is mandated, is a regulatory mandated. In fact, the availability of protocols is really important for transparency, but also for exchanging key information across different researchers and academia and regulatory bodies. Studies are often sponsored by pharmaceutical companies, and the ones that are registered irrespective of whether are imposed by regulatory agencies. Much more focus should be given to the multiple database studies, and also will be able in short time to also have a better understanding of the impact in terms of adoption of regulatory outcomes in relation to the models that have been used. This is a key point, especially I mean, taking into account an European perspective, not necessarily is the same as what we observed in the US with Sentinel. I'd like to thank you for the attention. I thank again all the partners that have been actively collaborating in the context of the working group 3. Happy to take any question if any.
Thank you very much, Gianluca. Any burning questions for Gianluca? You want to think a bit more? I think we're going to have good time at the end of the meeting. So over to our next speaker, Yeti Overberg from the Pharma Institute, where she works as a senior research manager. And she's going to talk about experience from a Ncep research center on the topic of how can Ncep contribute to research. Over to you, Yeti.
Yes, thank you. And good morning, everyone. Can everybody see my slides?
Yes.
Yes.
So my name is Jetty Overbeek. I'm a senior researcher and epidemiologist at the Pharma Institute, and I have no further conflict of interest to declare. So first, I would like to start with a bit of background regarding the Pharma Institute and our network and our main activities. So the Pharma Institute is an independent scientific research organization dedicated to the study of pharmacoepidemiology. And we have access to the Pharma Database Network, which is a longitudinal, large and high quality database network with real patient data from various sources linked on a patient level. And we work closely with national and international medical universities and European healthcare database partners. And examples of such close collaborations is through the performance of the post-approval safety studies and the post-approval efficacy studies. And since 2011, Pharma is registered as a research center in the Ncep. And our first collaboration in a PAS study was in 2012. And in 2013, we did our first coordination of a PAS study. So being registered as a research center and understanding the available tools that Ncep provides some advantages for us, and especially the EU PAS register and the Ncep resources database have been very valuable tools. So to start with the EU PAS register, we use this in several ways. First of all, we ensure to register our multi-database studies, especially the ones we coordinate. And hereby we increase the transparency of ongoing scientific research. Second, we also use the EU PAS register to go through the protocols and results that are publicly available, just to make sure we are aligned and we can use it as information when we design new studies within Pharma. And another way we employ the register is as a tool for business intelligence, because it also gives us an idea which PAS studies are currently ongoing and which research centers are involved with them. So for instance, in case of an ongoing PAS for which Dutch data was used and we are not collaborating on this, we might like to know which institution was involved and whether there would be any possibility to collaborate or to link with them as well. And also the Nsap resources database has been very valuable because this information is also publicly available. We could easily get data or information regarding the network centers that are registered here. So during our first PAS study, we collaborated with the UK and Italy. So we had two partners, and having the information in the resources database available expanded our European network to 9 partners in 2021. So it's very helpful. And with the increase of our European partners, we also gained more experience in our approach for multi-database studies, which is now much more standardized. So when Pharma is coordinating a multi-database study, we like to approach this in this way. So we would like to start then with a feasibility assessment where we select the appropriate partners, and once they agreed, we start the contracting phase, and once that is all settled, we can move to the study setup, so phase 2, where we develop or review a protocol and also make sure that all our partners review the protocol and start getting approval for their ethical and scientific committees to participate in the study. And in the meantime, Pharma will also start working on input file instructions, which I will elaborate a bit further on the next slide. So once that's all in place, we move to phase 3, where the local partners start the extractions, and furthermore, Pharma makes sure that common scripts are sent to the partners so that benchmarking results can be provided to Pharma to make sure that we can already detect issues with the input files prior to the execution of the actual program. And then we can start with the full execution once the input files seem all correct. And we can execute the actual study with common scripts, and Pharma provides then an analytic support if needed. And once all the common scripts have been run and aggregated data is sent to Pharma, we can start with the reporting phase. So when Pharma is coordinating a multi-database study, we use a common data model with study-specific data. So in this approach, data intended to be used for the study is locally extracted and loaded into the common data model. So that's all stays at the local database site. And then it's processed also locally with use of the common data model that has been shared by Pharma. And then the output is then sent to Pharma, which is usually at an aggregated level. And the advantage of this approach is that partners can require that the type of data to be shared complies with their local obligations and laws. And furthermore, data processing quality control is very high because partners are involved in the extraction, conversion, and transformation. And they have the knowledge of their data, so we believe that's the best approach usually in general. And also the scientific independence of the local researchers is high when using this approach. So this is what we usually do within Pharma, but we also partner, we're also collaborating a lot in multi-database studies. And by doing so, we have also gained many more research experience by having more discussions with our partners and really giving the quality of the studies improving. And this is also seen in the track record of our multi-database studies. So we see that, for instance, prior to 2017, approximately 20% of our studies we coordinated a multi-database study, or it was a collaboration for a multi-database study, and this really increased over time to over 60% in 2019. So as I explained, the EU Pass Register and these resources database have been really valuable tools for Pharma. It helped us to identify potential high-quality partners, but also further transparent our scientific research. And furthermore, the checklist for protocols that Francesca already mentioned is also very often used within Pharma to make sure that we cover all sections for a protocol appropriately. And also recently, Pharma introduced an internal meeting in which we each time discuss new studies, and especially the methodology, and the methodological guide will also be used during these meetings to discuss further and make sure everybody is aligned, and also the new colleagues know how this works. However, there are also some tools that are less used with it from our experience. So we, for instance, for the code of conduct, we do appreciate that it's there, and that we can refer to it. But and we of course adhere to it, but I think we could read it more often to make sure we have all updates still in our heads. And also we believe that these documentation would speak clear to us if we were to be involved right when there's an update taking place, so that it's more interactive. And for the Ncep study seal, we do note that we do not really aim for an Ncep seal by default for every study we conduct. Mainly, this has been done when our client really requests us to do so. So to conclude this presentation, we came up with a number of recommendations that we believe would increase the usefulness of these tools for Pharma and maybe also organizations similar to Pharma. So we would appreciate more interaction and more involvement from research centers when documents are developed and/or updates. And as the number of research centers is really increasing, we thought it might be helpful to have kind of ranking for the research centers, for instance, regarding experience, to have an easier search. And also it might be helpful for us to have more knowledge or information about the Ncep seal to maybe increase the willingness to aim for such a seal. So that was the last slide regarding our experience with the Ncep tools. And if anybody has a question, I'm happy to take these.
Hi, Susanna, this is Arnold. I'm here.
Yes, it's a great pleasure to introduce Arnold Chan. He's the director of the Health Data Research Center at the National Taiwan University in Taiwan. And we have him here also in the steering committee. He represents the International Society of Pharmacoepidemiology, ESPE, and he's going to share some experiences on how Ncep can contribute to research from a non-EU academic perspective.
Thank you, Susanna. This is Arnold. I am from Asia, the farthest away from Europe as possible at this point. And I'm really pleased to have a chance to share this, to share some thoughts with you. May I go have the next slide, please? So I do not have a conflict of interest to disclose. And next slide, please. As Susanna mentioned, that I represent ISPE, and really an honor to participate in the Ncep steering group. I do have much experience outside of Europe, mainly in United States and East Asia. So in my professional career, I have been doing similar things that my colleagues have been doing in Europe. So today I'm just going to share with you some thoughts from outside of Europe. May I have the next slide, please? ESPE, many of you are well known to it, and you're most welcome to log on and to find out the latest development. The next speaker actually is going to chair, the mid-year session, very soon. So I will not repeat that. And basically, ESPE, would like to work with scientists across the world to carry out high-quality studies, with public health and regulatory impact. And I think from this perspective, ESPE and Ncep is very consistent with each other. And in fact, the key players are mainly, there's much overlap between them. Olaf is actually the current president of ESPE. May I have the next slide, please? So today, I'm not going to use up my full 10 minutes, but I just want to highlight, from outside of Europe perspective, what I personally view as, three major points. Because we work with commercial products, and there's so much potential conflict of interest involved, therefore a transparent and independent code of conduct is crucial. In the Western world, so to speak, United States, Canada, Western Europe, it's codified. And but in other parts of the world, we are in different stages, of implementing these practices. So therefore, having a written code of conduct, under Nsap, which is like endorsed by the EMA, is very important for us, outside of Europe. There's another example that we can cite, such that, of course, we work under different laws and different history and different culture, but, having this in a very explicit manner, I can never overemphasize, how important it is. So that's the most important thing. And then there's a couple of other points I will describe in the next slides. May I have the next slide, please? So this slide have already been revealed in a previous presentation, so I'm not going into the detail. But I would highlight the fact that for me as a educator and also as a researcher, I am very pleasantly not surprised, but I always pleasantly find that this methodology page, is frequently updated. Just to give you an example, Some years ago, two other people and myself edited a book called Pharmacoepidemiology and Therapeutic Risk Management. It was published in 2008. And at that time, some of this content, were up to date and state of the art. But as the field evolves, there are new regulations, there are new scientific methods being developed. It's critical to update that. But from a book setting, it takes a lot of work. The turnaround time is much slower. So therefore, having a always up-to-date methodology guidance is very, very, very important. And from my own experience, I basically use it, as a teaching tool. So in addition to asking my students to read textbooks, I actually refer them to this methodology guidance document so that they can find out the latest, the case crossover design, the self-controlled case series, the large pragmatic trials and things like that. They're all here. So this is very important, globally, and not just in Europe. So may I have the next slide, please? And the previous speaker also discussed, developing data across multiple data sources. I personally have some experience when I work in the United States. So I was involved in the development before the US FDA Sentinel. So I know what is involved in developing consistent data across different data sources. And in recent years, in the recent decade or so, there have been similar development in East Asia, not across all of Asia yet, but Korea, Japan, Taiwan, Hong Kong, Singapore, Australia, there have been strong interest to develop some capacity similar to Nsap and similar to the Canadian EndNotes, and therefore, all the best practices development in under Nsap, those are extremely helpful as well. May I have the next slide, please? So this is the last slide. Like many of you, many of us in Asia are working on exactly this topic. We are slower. Most of the countries in Asia are slower than North American and Western Europe in introducing the vaccines. But for example, Koreans started vaccination, I believe last week or a week or maybe 10 days ago, and so is Japan. So we are basically following the same game plan as in Europe, and very much appreciate the recently published AESI incident rates under the Ncep website, and actually just to show my appreciation and just to show how relevant it is, our CDC officers, in Taiwan, quickly pick up that, and we're engaging in very in-depth discussion. And of course, the incidence rate probably different because of the different ethnicity and genetic makeup. But just the methodology, the transparency of the codes being used, the ICD-9, ICD-10, and all that, those are extremely helpful. And as we move forward with more exposure, I do believe that there will be more room for discussion, and we look forward to learning from each other. So this is my presentation, and I thank you very much for your attention.
Thank you very much, Arnold, and thank you also for staying for the Q&A part of this webinar.
I will.
Yes. And our next speaker is Helga Gardarsdottir. She is a professor at the Division of Pharmacoepidemiology and Clinical Pharmacology of the Utrecht University. And she will be sharing considerations on COVID-19. This is the first of two COVID-19 dedicated presentations today. This one is on good practice in observational research in COVID-19. Over to you, Helga.
Thank you, Susanna. All right, thank you, Susanna, and thank you for inviting me to offer some reflections this morning on considerations of good practices in observational research on COVID-19. And next slide, please. I guess it's difficult to start such a talk without putting the topic in perspective. And although I'm sure that all of you are aware of the storm of debates and discussions involving the use of real-world data, high time urgency to answer questions, to kind of support regulatory and clinical decision making during the pandemic. We just year, I think, ago when this all emerged. And at that time, we didn't know exactly how to react and what do we do at that point in time. We do what we do best, and that is to look at the data, try to understand it, and generate useful evidence for clinicians and policymakers. There were at that time abundance of hypotheses and rumors about specific medications that might be useful or harmful in patients that got infected with COVID-19. And although I'm not going to enlist them all here, I just put on this slide the hydroxychloroquine, chloroquine, as well as the ACE inhibitors, which were both put into context when it concerned COVID-19. Next slide, please. Next slide. Yeah. Oh, then everything comes in abundance now. Well, this quick generation and high pressure of timely publication of evidence, including quick reviewing in prestigious journals, did not always turn out well. And I'm assuming that the Surgisphere study is now known to all, although I think it's all clustered in this slide. I was intending to show this multinational registry study that assessed the effectiveness of hydroxychloroquine and chloroquine in treatment of COVID-19 patients. And it was a really large study with, I think, about 96,000 patients, resulting in showing that actually these therapies were harmful to patients. And this study actually had a huge impact, even leading to suspending of clinical trials by the WHO in different countries. However, after vigorous review by the research community, a number of inconsistencies were identified in the data originating from this study, which actually included a research network called Surgisphere, which was not really known to many, also surprising to us within the pharmacoepi community that none had heard of this large data source before. And I guess many of you know how this ended, as authors were unable to provide information about the data. All these inconsistencies indicated some sort of falsification of the data used, and in the end, the article was retracted. But I think it shows at this point in time what a huge impact the evidence that we generate can have on clinical practice. And although this example is kind of extreme, and I'm not sure if many others like this occurred during that period, there were many studies that showed methodological flaws. And I will provide one such example later on. These malpractices and flaws bring into light, the importance of good practice when we're performing observational research. And I think since then, at least 2 papers have been published that provide guidances on performing observational research. Next slide, please. The first one is actually from our colleagues within Ncep, and it was published quite early on. It was published in April, I think. And just recently, our colleagues in the US also published a study where they also specifically discuss assessing vaccines and vaccine safety and effectiveness and what we should consider for such type of studies. So the references are included there, and I really encourage you to read these 2 because they really provide good guidance and insights for us when we are designing these studies. Next slide, please. I've just put up here what Pottegaard and colleagues put forward, the considerations that they thought of when it regarded performing pharmacological analysis during the pandemic. And many of those overlap with what is reported by Sneelijs and colleagues. And although I only have 15 minutes and I will not go into all the details, I put them all on this slide, and I just want to say a few words about these different aspects. So if we start with, for example, timeliness that they identified, this was an aspect very important during the first peak of the pandemic, and maybe still is, as what we know, the high need for evidence, balancing that with resources that are limited. There still needs to be some sort of selection that the urgent important questions should be prioritized when we are thinking about conducting these studies. Observational studies on efficacy, I think that's also very important, that when we are performing these studies, that we clearly state the explanatory nature of such analysis, and that these are able to provide hypotheses on promising drug candidates, but really keep in mind that the golden standard for efficacy studies is the randomized controlled trial. So that's good to keep in mind when we are also reporting on the results from the studies that we perform. Implementing real-time epidemiology, this is of course a topic that we've been working on for many years, and I think specifically during COVID it was very challenging, and it is in general challenging because we have lag times in data availability. I think data registration and collection was probably very different in these overwhelming healthcare systems during the time of COVID. So when we embark on designing studies, we should really work closely with data registry or database holders to understand the structure of these databases. And also alongside that, close collaboration with clinicians that really understand the decision making during the pandemic and can explain possible non-expected patterns that we see in care provision in the data, that we know how to interpret these and how to account for these in our analysis. The choice of study design will, of course, ultimately depend on the study question and the data availability that we have. That's always the case. But I think specifically for COVID-19 studies, an important aspect is identifying the underlying source population during a period of time when policies and availability of testing changed rapidly. How to identify COVID positive patients? As we know, throughout the past years, the frequency of testing, the availability of testing has changed enormously. Also, assessment of exposure, one of the cornerstones of pharmacoepidemiology. We have a long-standing experience with using outpatient dispensing or prescribing data for exposure assessment, while many of the COVID patients were treated in hospital. Unfortunately, we do not often have the same level of access to exposure information in hospital, and sometimes if we have, that registry or database might not necessarily register what is used prior to admission, which is information that we need to be able to account for confounding or take into consideration other drugs that might also affect the outcome that we are looking at. And also another aspect is that prescribing practices were gravely impacted and indications were dissolving during this period, bringing new challenges when we're designing our studies. And I will come back later in my presentation with a small example of how ascertainment of exposure during COVID actually went wrong. Likewise is the selection of covariates that we use to describe population characteristics or to handle confounding. For COVID studies specifically, there's a challenge because we lack information on risk factors for infection, and we also don't have the unmeasured individual characteristics such as adherence to quarantine regulations that are likely to play a large role. Also, many covariates, such as severity of illness and laboratory findings, could potentially be intermediates in the causal pathway between pre-admission use of drugs of interest and the outcome of COVID. So careful consideration is needed when we consider what to be included in our analysis. defining the outcome, that is also usually a challenge, but as said, and also relate to the source population during a period with varying test strategies. So we need to account for these changes and differences in COVID testing between countries as well as within countries and over time. And we have various means to try to do so, such as applying stratification or matching on place and time, but really important to have this in consideration when we are designing our studies. And I think lastly, the transparent reporting, and this has also been mentioned by prior speakers, it is key. We need to assure that we adhere to existing guidelines from the Ncep guide on methodological standards was mentioned, and I know other global organizations such as ISPI also has produced good pharmacoepidemiological practices guidelines. It's important, and we should register our study protocols so they are openly accessible, just as mentioned in the EU PASS register, to facilitate the quick and efficient assessment of new evidence, as well as providing opportunities to perform replication of these studies that are sometimes needed. I mentioned that exposure assessment is the cornerstone of pharmacoepidemiology, and I just want to provide an example on how that actually went wrong during COVID. And for this, I've chosen a study by Zhang et al. Next slide, please. Published in April 2020. And you might be familiar with this study, but it assessed the association of inpatient use of ACE inhibitors and ARBs with mortality in hospitalized COVID patients. And they reported a significantly lower mortality in patients that use these differences. Next slide, please. However, upon further assessment, how they designed the study. You see here, many other studies followed this that showed no association, also from colleagues within our network. So it just shows that there is controversial evidence out there, and we also know that what we put out there also impacts how clinicians actually treat their patients in clinical practice. So there's a high responsibility of performing these studies in a good way. And if we look just at this example, which I wanted to show here, if we look at the assessment of how they designed their study, specifically how they dealt with exposure, there were some methodological flaws identified. And these flaws are not only seen in this particular study, also in others. Next slide, please. And I've just put on the screen here a copy of how they defined their exposure as in usual pharmacoepidemiological research. We compare the cohorts of those that are exposed to those that are not exposed to the therapy that we are interested in. And we see here that in this case, it was that people receiving a certain drug during hospitalization were defined as users and they were not users. And I've just included an example there on the right, just to see that this is not an example on its own, but more studies have performed it in this way. And what's the danger of doing so? Next slide, please. Well, it's figuratively presented here. We see these two cohorts of patients that kind of define use during hospitalization as being exposed without thinking about when actually the exposure started during hospitalization. So we have this dotted line there that shows time that the patient is not really using the drug, which is actually time that the outcome should not be able to be allocated to the exposure of the drug. So next, you push the next one, it should start moving. So, back, please. Well, the effects don't seem to be working, because what I was supposed to show you now is how this dotted line should actually move to the second cohort there, where there's no use, because the time that has been allocated there should, of course, not be counted as exposed time, as it creates this immortal time bias in pharmacoepidemiological studies. And this is has been shown in many of the in-hospital drug use studies that have been performed to assess influence of certain use of drugs on COVID-related outcomes. And next slide, please. Yeah, for those of you interested in learning more about that, there's recently a publication by Sami Svisa and his group on the possible biases, also showing how studies applied this in hospital exposure to define their cohorts and what the impact of that actually was on study outcomes. So this is just an example of how actually methodological flaws can lead to biased results. Next slide, please. And while we've been discussing with you considerations when it concerns good practice for observational research on COVID-19, we should not forget to think about what the pandemic means for future observational studies, including those on COVID-19. There was recently a letter to the editor in PDS by Webster Clark that reflected on this, and I thought they were very interesting and very relevant for our Ncep community. How have our databases been affected by COVID-19? I mean, we will have heterogeneity and representativeness, and although in Europe we have databases with national coverages where this might not play as big a role or different role for claims databases, we would expect non-random dropouts, maybe possibly related to socioeconomic status. We've also seen differences in healthcare usage throughout the pandemic, which will be shown in the databases that we use. We also can expect that the patients are going to be impacted in the way they use medicines, because there was much reporting on what was beneficial, what was not beneficial, what was harmful. And patients also make their own decisions on how they use their medicines. So that's also something that we need to account for in future studies. Next slide, please. And just pulling up here, we have, of course, these data, we use them to identify covariates and confounders outcomes. And if we see here, how will this work in the future when we have this unstatic healthcare utilization? During COVID, we saw a decreased incidence of diagnosis, less referrals. Next slide, please. We saw also, for example, here in these cases, up to 90% less screening for breast cancer, 85% for colon cancer. And the next slide, please. And I think also, as mentioned, various changes in prescribing patterns, less initiation, peaks in certain type of use at certain point in times. So next slide, please. So for this slide, I think I put, what's the value of being a member of Ncep? Also for the future, I think we have lots of challenges, although we have now provided and are providing guidance on how to perform good pharmacoepidemiological research with our methods guide that is continuously updated, also accounting for issues that we need to think about when we're performing COVID-19 studies. I think, being a member of Ncep is that having access to this collective knowledge, there's so rich knowledge and experience within this community, continue to learn from our ongoing and future multinational collaborations, and assuring that we continue to provide guidance to the research community and publish standards that help others to comply with good practice for conducting observational research. And I think most importantly, to spread and to continue advocating for and providing means to be transparent, stimulate registration of observational research in, for example, the EU PASS register. And I think it's a real value to be a part of that community, to learn and also to be able to share experiences within that community. And I think in the end, it all boils down to continue campaigning for the careful consideration of pharmacoepidemiological principles. And I think with these words, I would like to end my presentation. Thank you.
Thank you very much. It was very clear and very interesting. Is there any burning question for Helga now? No one wants to ask question today. Okay, we leave it for the end. And now we go to the next talk. As mentioned before, there are two talks on Ncep and COVID-19. The next talk will focus on how Ncep can contribute to the monitoring of therapeutics and vaccine, and it will be presented by Danny Prietro, that is a professor of pharmaco and device epidemiology at the University of Oxford. Thank you very much, Danny.
Thank you, Jean-Mario. Yes, I'm delighted to be speaking to you all today on this topic. Next slide, please. I'm going to cover these 4 bullet points in the next 15 minutes. I'm going to be talking about distributed networks and how we build from the community and work together to create knowledge and evidence rapidly, and how we have done that in the last year. I will then cover the eCore network that has been created to look at the safety and effectiveness of COVID-19 therapies in the future. I will cover briefly, and I will start by thanking Helga for her knowledge and slides, access and the work that has been done on vaccines so far. And I will close up maybe by reinforcing the importance of international collaboration on COVID-19 research. Next slide, please. Next. Yeah, so just very briefly, and this has been said already, the way we are collaborating and the way we have collaborated and produced evidence in the last year using common data models has been, I believe, quite interesting. There has been an increased uptake of common data models globally, which has facilitated the analysis of patient-level data without moving patient-level data, basically through the mapping to a common data model that can then accommodate this common analytics. Next slide, please. The advantage of this approach is that we can, next slide, please, we can break down this kind of convoluted journey that goes from patient-level data to the provision or the generation of reliable evidence by creating first a patient-level data, common data model version of the data sets, and then breaking down the tasks into standardization and analysis execution. And by doing this, we make it possible to reuse and reprogram and recycle and improve data curation and analytical code. Next slide, please. I will now cover the eCore network and what it means for Ncep and future work on COVID-19 medicines. Next slide. eCore is an EMA-attended network for multi-center collaboration for COVID-19 pharmacoepidemiological research, and it stands for European COVID-19 Observational Research Exchange. Next slide, please. The why we need this is somewhat obvious, I guess, to people in the audience, but maybe not so much to people who mostly run randomized controlled trials. There are indeed many randomized controlled trials ongoing, many pragmatic trials with relatively large sample sizes. But despite all those efforts, which are indeed very welcomed, we are going to be left if we don't do any pharmacoepi evidence or research without data comparing different treatments. And we would be left out without knowing, for instance, whether all corticosteroids are equally safe or effective, or whether anticoagulants are better than antiaggregants or platelet inhibitors. Next slide, please. So the overarching aim of the eCore network is basically to create a framework to support collaborations for multi-center international observational studies on the utilization, safety, and effectiveness of medicines used by COVID-19 patients or to treat COVID-19 patients. Next slide, please. The why I have said already, the what, the eCore network will deliver 3 things: a library of cohorts of COVID-19 from around Europe, a collaborative framework across the network to support the use of this library, and finally, a large multinational pilot study that will be used to test the system that has been put in place. The initial network will cover a total of 7 European countries. It will be finalized and up and running by June this year, and it's inspired and has learned a lot from previous collaboration between the Eden and Odyssey communities in the last year and all the work that has been done on COVID-19. Next slide, please. These are the milestones and deliverables. There is a protocol for a proof of concept study that I will cover in just a second, a governance model of the network and a protocol template. We will, in the coming few weeks, have study results, and by the end of the grant, so by June, we will do a review of the efficiency of the network and look into the future to see how this can be sustained. Next slide, please. Again, the idea here is to use data from many European countries, but potentially engage with colleagues in other parts of the world to map data to a common data model, and once mapped, then analyze that data without moving patient-level data beyond borders. Next slide, please. The idea is to have data from all these data sources. So far, we have data from hospitals from Spain and from primary care records and ambulatory records from Belgium, France, Germany, the UK, Italy, the Netherlands, and again, Spain. There are hospital EMR databases that are being mapped, and data also from Serbia and Germany that are being contracted and mapped so that we can use them for the pilot study that I will cover next. Next slide, please. This is just a screenshot of the data characterization dashboards that we have created, which is basically an interactive web application where one can see the characteristics of the patient involved in the network. To the right, you can see the age representation of data from France, Germany, Italy, Netherlands, Spain. And again, Spain, the last one in green to the bottom right, called HM hospitals, is hospitals from Spain, and you can see how the social demographics of the age distribution is skewed because as we know, hospitals tend to admit older patients than primary care records. And to the left, you can see a little bit of a flow chart on how patients are diagnosed, tested, and how we collect data on hospital admissions in one particular database, in this case, the CDAP database that covers about 80% of the Catalan population. Next slide, please. The proof of concept study is basically a drug utilization study looking at the use of systemic glucocorticoids or corticosteroids in COVID-19, which will also look at the risk of adverse outcomes and complications in the first 6 months following COVID diagnosis in people treated with systemic glucocorticoids, starting from a kind of ambulatory perspective and also an inpatient care perspective, including 7 countries, as I mentioned earlier. And if we go to the next slide, please. And including these different definitions of COVID-19. COVID-19 is a condition that, as you all know, can be tested with a number of tests, typically real-time PCR testing. In the first wave in Europe, which was mainly in Q1, Q2 of 2020, there was lack of testing capacity, which results in many patients being diagnosed without having been tested. And we are going to be including these different definitions and different cohorts in the analysis to see what is the impact of different phenotypes to identify COVID-19 patients, including diagnosis based on a clinical diagnosis, diagnosis based on a laboratory diagnosis, diagnosis based on symptoms, or diagnosis based on a hospital admission, or the most inclusive, which is basically any of the above. Next slide, please. We will look at prevalent as well as naive or new users of corticosteroids for the drug utilization study, and we will be looking at what happens to patients when they are using corticosteroids after being diagnosed and after being admitted to hospital with COVID-19. Next slide, please. I'll now cover very briefly the access network on vaccines. Next slide. Again, I'm going to thank Helga for sharing some slides and some thoughts on this. This is now again attended network interested or created to do real-world monitoring of COVID-19 vaccines. Next slide, please. There's a number of partners involved in this great collaboration, including the leadership of Professor Stolkenbaum, who I think is in the call, and Professor Klungel, and many of the partners that have been presenting previously. And I will not read all of them because it's a long list, but you can see the logos here. Next slide, please. The idea is to create readiness for vaccine safety monitoring, to create event definitions, codes, and algorithms to identify and to measure the risk of adverse events of special interest that have been prioritized, to create common protocols and tools, and to identify data sources that are fit for purpose for the necessary research. And this will include the estimation of background rates of these adverse events and post-marketing research, including safety assessment, effectiveness, coverage monitoring, and integration of benefit and risk evidence. And the first bit of data that we have seen so far is the generation of background rates for these adverse events in, I believe, for now 3 countries, but I also know that more data is coming up to include a total of 7 countries in the coming weeks. Next slide, please. These are the databases that we know are involved in this network so far, including data from Germany, the Netherlands, Denmark, Spain, Italy, the UK, Norway, and France, and a total of over 138 million people. Next slide, please. I've taken the liberty to depict the data that has been put in the public domain by the team, and you can see here a little tableau representation of the common adverse events of special interest, or at least 3 of them, just for illustrative purposes. I just want to mention from this that one thing that one can learn very quickly, only looking at this data, is that the old idea of having one incidence rate per event, per country is probably not going to work, because you will need to take into account, as expected, of course, age, because there is a very clear trend with age, at least for these common events. And this is data, sorry, I forgot to mention, from 3 countries and 5 databases. Next slide, please. This is now a representation of the rare adverse events, and the only thing I will mention here is that it seems obvious that there is more variability in the measurement of incidence rates when one looks at rare events compared to the most common events, and that could be, of course, a function of precision. But it's also clear that the age trends are not necessarily the same for all of these events, and therefore it is, again, going to be very important that for all vaccine safety research, we take into account age as a factor that needs to be considered, including in pharmacovigilance and the collection of events, of reports. Next slide, please. And finally, just to show, again, very rare adverse events, if one focuses maybe just on the left one, encephalomyelitis, great variability in the measurement of rates of these events, probably due to a lower power, statistical power, and if one was to plot confidence intervals, we would probably see that. But I just want to mention that that of course it is very important that we use consistent definitions, and that we look into the definitions of these events. And I think it's very important, the work that's been done on the creation of algorithms and good code lists for the identification of these events. Next slide, please. I will close by mentioning some work that we've done in collaboration with colleagues even beyond Europe. And I will start maybe in the next slide by covering very briefly the Eden and Odyssey collaboration on COVID-19 research in the last year. Next slide, please. This is a screenshot from a paper that's in Research Square preprints, looking at the network of cohorts that has been generated so far, including more than 4.5 million people tested positive and more than 1.2 million people hospitalized with COVID-19. Data mapped to the common data model from 9 European countries, as you can see there, and 13 US and 3 Asian nodes from China and South Korea. Next slide, please. And hopefully in this screenshot, you can see the social demographics. This is basically age and gender amongst the diagnosed, hospitalized, and those admitted to ICU in the different data sources. And you can see how in red, most of the people diagnosed with COVID-19 are in the young adult ages, while most of the hospital admissions in blue happen in people in their 50s, 60s, 70s, and most ICU admissions more in the 50s, 60s, and less so in the 70s or 80s, probably because those people are not fit for purpose for ICU admission. Next slide, please. This is now an addition of additional data from the Eden Odyssey collaboration, looking at the background rates of adverse events of special interest. I've basically merged this with the previous ones for illustrative purposes here. And I think this is a great example of how our communities can rally together to generate very important data on the background rates of common adverse events of special interest for vaccine safety using data from all the databases that Access has generated already, and the addition of a few additional databases from the US, a few European countries, and I think a database from Japan as well, JMDC, which is claims from Japan. Next slide, please. And just an example of how the data from the mapped version of CPID Gold maps with or matches the data from the Access estimates, at least for the narrow definition of these events. And I think you can see how there is at the bottom of these plots in purple or maroon, and in orange, the comparison of both CPID Gold estimates that are very close together. Next slide, please. And finally, to finalize, I'll just cover very briefly some work that's just beginning now on a coagulopathy, COVID-19 coagulopathy work that we are leading from Rotterdam, Oxford, CDAP, and IQVIA. This is a collaboration that is being led from the EMA perspective by us, but then by colleagues from the American continent by the FDA and Health Canada. Next slide, please. The research question and objectives is relatively simple. We are going to be estimating the incidence of venous thromboembolism in patients with COVID-19 at 1, 2, and 3 months after diagnosis. We will then calculate the risks of worsening once someone has COVID-19 after having a venous thromboembolic event. We will then look at the impact of risk factors of VTE among patients with COVID-19, and we will end by developing and externally validating a patient-level prediction model to identify people at high risk of venous thromboembolism amongst those with COVID-19, which could, of course, help us manage this condition differently. Next slide, please. And to finalize, the same basically, but now for arterial thromboembolic events, we will estimate incidence rates, calculate the risks of worsening COVID-19 after having one of these arterial events, look at the impact of the known risk factors on these rates, and finally develop and validate patient-level prediction models for arterial thromboembolic events targeted for patients with COVID-19. This is my last slides, and I will finish opening for questions and comments. Thank you.
Thank you very much, Danny. Any burning questions for Danny, or we reserve everything for the Q&A? Okay. Well, it's Monday morning. Thank you very much, everyone. I think in the interest of time, because we want to finish at noon, Central European time, maybe, Gianmario, we can streamline a little bit our presentation here. And the goal of this is that Gianmario and I, as who have the privilege to co-chair the Ncep steering committee, give you an overview, next slide, of what have been important. Can you move to the next slides, please? Thank you. What have been important achievements of Ncep? Why is a new Ncep mandate needed? And what are these elements of the new Ncep mandate? Next. I think we have a very mixed audience today. I know there's some of the old timers that were part of the network already from the very beginnings in 2007, and some that are more recent members of the network, and some that probably are exploring whether you want to become members and what's in there for you and collaborate with others. I don't want to dwell into what have been the important achievements of Ncep. I think we've heard about it. I think a key element is the guide on methodological standards in pharmacoepidemiology, very much aligned and using as a jumping board the ESPE good practice guidance related to this. This working group has been adding additional topics, most recently measure the impact of pharmacovigilance in some of in new sections. And as it has been highlighted, an important element is that this is a working group that works, is this a guidance that gets updated every year. So everybody's welcome to contribute in this working group as well as in the others. Also, the working group that has updated the code of conduct has been working relentless, addressing some of the queries from other groups, and including external stakeholders. A lot of work on clarifying the best practice in relationship between investigators and study funder. Clearly, there's been a lot of work into making information accessible through to facilitate transparency of observational post-authorization studies in the EU PAS registers, notably, but also through the other tools that have been covered earlier, so that we know who is working on what networks are already out there, what data sources. Not on this slide, but I think it's very important, and this was very important, especially in the early days, but hopefully as the network grows, we'll be able to go back to the face-to-face meetings that were so instrumental in the early days to get us working together above and beyond the interactions that we already have through societies like ESPE or ISOP. So it has been really instrumental in developing all these networks to do all these activities that we have been sharing before. Next, the outgoing steering group has identified. Sorry, can we go to the next slide, please? Thank you. The outgoing steering committee identified a number of aspects not fully achieved, or new challenges and opportunities. And among the aspects not fully achieved is that the suggestion that we improve the communication and the dissemination of Ncep outputs and the promotion of educational activities, that the methods recommendations get disseminated and integrated to recommendations for national medicines agencies, and also particularly on the technical side, improvements in the EU PAS register and the resource database, and also not only in the technical, but thinking about how real-world evidence is expanding and moving beyond our traditional fields of pharmacoepidemiology and pharmacovigilance, that make sure that we can leverage what we do in those networks. Next slide is the one on new challenges and opportunities. Obviously, as mentioned, there's a lot more applications that are being very visible to everyone in the area of real-world evidence, new data sources, new approaches, and fields like artificial intelligence, obviously, and all the expectations on leverage all this data so that we can provide knowledge and evidence for decision-making to many stakeholders. COVID-19 obviously is one of the key priorities and has been a key priority for our community over this last year, will continue to be, and a number of international digital digital initiatives, and going from specifically the health heads of medicines and EMA joint big data task force report on how to establish a sustainable platform to access and analyze healthcare data from across the EU, which is driving already a number of projects in this field. but also the legislation to create a European health data space, and that will come at the end of the year. And in 2020, the adoption by the Commission of a digital strategy and artificial intelligence white paper. And finally, these are all areas in which there's a lot of opportunity, as Helga showcased. What we do with data, especially when the data are out there, as in how important it is to apply rigorous research methods and collaborations, as Danny also pointed out. And moving to the next slide, another aspect is to make sure that we involve and we keep in mind the perspectives of stakeholders. Make sure that the guidance is particularly focused on the accessibility and suitability of data for data sources for regulatory purposes, to make sure that we leverage the expertise and experience of investigators in industry while safeguarding research independence, focusing on the scientific collaborations. And then the International Society of Pharmacoepidemiology and also the International Society of Pharmacovigilance have been traditional professional learned societies that have inspired a lot of the members and were a lot of us working, but also keep in mind, again, in the context of real world evidence, how many fields are getting closer to the kind of work that we do, and make sure that we also collaborate and keep in mind the perspective of learned societies like ISPOR, but also other bodies like health technology assessment, payers. An important aspect is also the disease registry networks. Over to Gian Mario.
Thank you very much, Susanna. So we have seen what Ncep has achieved, thanks to Susanna and the other presenter. We have seen why a new Ncep mandate is needed. So now let's focus the last 5 minutes on the future. And I would like to thank Xavier and the other member of the previous steering group for providing these elements for the new mandate. So most important to say, the new mandate and the new elements will be at the core of the Ncep mission to strengthen the monitoring of the benefit-risk balance of medicine. So they will serve the core mission of Ncep. And we have identified 4 main elements. The first 3, they all aim to have high-quality studies. How? Well, first, having access to data. Ncep will continue and increase the support to the identification and access of high-quality data. Then, once we have the data, we need to develop and use a methodological standard and governance principle. And finally, we need to bring together capacity and expertise across Europe, what Ncep is doing. And all these 3 elements put together will help to have high-quality studies. The 4th element is new for Ncep and reflects what's happening with the current pandemic. We have seen that real-world evidence has informed the response to the pandemic. We have seen how real-world evidence can be important in answering the question the regulator have around how to manage, to handle the pandemic. So we would like to leverage this network of excellence in a way that it can be used to deal with this and future public health emergencies. Finally, and as a note, as Xavier already mentioned in his presentation, just a reminder that Ncep will continue not to perform regulatory activity related to specific substances, products, or class of product, and will not provide a forum for the assessment of their benefit. So now let's go slightly more in detail of each of the 4 elements, starting with the access to data. So what are we going to do in this area? We are going to do mainly 3 things. The first one is that we are going to support data discoverability, both in terms of identifying the data sources that are adequate for research and decision making, but also in develop a standard of set of metadata that will help to describe these data sources so that we can know what characteristics there are and which data source is more relevant for a specific research question. Once we have this information, we can then contribute to the development of the data quality framework and apply this framework to the data sources. So we will know even which of them provide high quality information that are relevant to a specific research question. In doing these two activities, we are also promoting the FAIR principle, so to make data sources more findable, accessible, interconnected between them, and using them more often. Then the second element was about methods and governance, and even here we foresee three main activities. The first one is that we want to continue to develop and update guidance to support good methodological and governance principles. So we want to update the existing guidance, and these were mentioned many times today: the Ncep guide on methodological standard, the checklist for study protocol, the code of conduct. So we want to continue to update them, but we also want to develop new guidance as needed. Then, and this was mentioned as well before, we want to strengthen the collaboration, both with existing Ncep stakeholders, but also with new networks like the Darwin network or the registry network. And this will help to develop and promote the use good of methodological and governance principles. And finally, we have seen in the previous presentation how multi-database studies now represent the majority of studies. There is a high interest on this. So we want to strengthen the recommendation on this type of study from an analytical point of view. And this is currently being done by the Ncep Working Group 3, led by Gianluca, and we want this activity to be even more important. Moving to the third element, high-quality studies. Here we foresee 4 activities. The first one is we want to continue and even more leverage the expertise of the various centers that belong to Ncep, and in particular leverage the expertise of the centers that have experience in relatively new methods like genetic epidemiology or artificial intelligence that are now not well covered in the current Ncep guidance. Then we have mentioned this several times, is the start of today, the UPass register. So we want to support the upgrade, make it more user-friendly, make it easier to enter the information and extract the information so that we can address stakeholder needs for information. And the work done and presented by Gianluca will be helpful at this regard. And we want to continue to promote the use of best practice guidance that will be used during the design, conduct, and analysis of studies. Finally, but not last, we want also to support the provision of training curricula in pharmacoepidemiology. So we want to develop training in collaboration with other learning society. And this leads to the last topic, last element of the new mandate. The new element is that, as we saw, we want to increase the capacity for rapid studies, in particular, but not only in case of public health emergencies. And this is very important because we saw that regulator and decisions sometimes cannot just wait. So we need to develop tools, processes, and mechanisms to be able to provide high-quality results in a rapid time. So the last slides before, I know you will have questions now, so I don't want to take further time from it. Just one slide on the organizational work. This was touched at the beginning, but just to summarize that there are 2 main actors in Ncep. One is a steering group that gives more an orientation and define the objective and decide on the operational task of the network. And then there are the working groups and special interest groups that are more temporary in nature, that are in line with the objective defined by the steering group, and is where the real content is developed. We currently have 3 working groups active, one on standard and guidance, one on independency and transparency, and one on multi-source studies. And we have 1 special interest group that is about measuring the impact of pharmacovigilance activity. So this would be a good starting point for new members to join. And of course, new working group and the special interest group can also be established as appropriate according to new research interest or new objective. So this is all from us. And now we have 5 minutes left for questions. Let's see if we have the first volunteer of the day. Xavier?
Yes, thank you, Don Mario. Perhaps I have a question, perhaps not for myself, but which I anticipate some of the present of the audience would ask. When some members would like to participate in one of these working groups, are interested in these working groups, and or in any of the research projects that have been presented or that may then be established in the near future, what is the mechanism for collecting the information or expressing interest?
I may one. I mean, there's two aspects to this. Obviously, if anything that is Ncep volunteer work related, all the information about the working groups is on the website. But even if you don't find who you should go for every working group, to send a message to the Ncep Secretariat is probably the fastest way of sending your expression of interest, and the Ncep Secretariat is very efficient in channeling these questions to the appropriate working group chair and co-chair. I think for the research element of this is a little bit different, because that is not volunteer work. This is done through the funding mechanisms, even from IMI, EMA, the Commission, pharmaceutical companies. I think if you're totally new to the network, you may want to look at presentations. Also in the public website of Ncep, there is a documentation of all the presentations and all the plenaries. Look which groups are working in areas that you're interested in. Another way would be to go to the EU PASS register and then contact the PI center. Gian Mario or other members of the steering group, any other thoughts on Xavier questions?
Thanks, Susanna. I think you touch all the important points. The secretariat that is very efficient, and the UPass register where you can see all the contact point of the center register in Nsap. Thank you for your answer. Let's see if we have another question, another brave volunteer. No.
Maybe I throw one, if you don't mind.
Yeah.
There was, I think, one of the closing questions on Yeti's presentation is what is the value of the Ncep study seal? And maybe if I can share that with Xavier, because that's a question that I think most research centers get a lot. What is the value from a regulatory perspective for a study to go through the Ncep seal? So not only adherence to the research standard and the code of conduct, but also apply to the Ncep seal.
Thanks, Susanna. We have to realize that the Ncep seal means a commitment, and it's a strong commitment from all the participants to the study to follow the principles of the code of conduct and the best methodological standards. So even if there is no immediate impact on the acceptability of a study on regulators and so on, this provides a reassurance that indeed there has been some high-level standard that have been applied to the study. In addition, the seal also gives regulators some means for checking and verifying this type of commitment, because there are a number of declarations that need to be done in writing by the investigators in terms of the Ncep checklist for study protocols, declarations of interests, and so there is verification of the commitment that are being made by the investigators. So this provides really for us at least a high standard in terms of governance of the study. So of course it does not imply necessarily that the result will be any implication regarding the results, but at least in terms of governance, in terms of conduct of the study, this gives reassurance about the quality of the process that are being applied in the study.
Thank you very much. Any other? I know we're already 1 minute above the time. There's a question. From Christine Damas, Michelle, formerly there was a sick on medication and pregnancy. What about this?
Yes, thanks for the question. He hasn't been active for the last month, and we are thinking whether to keep it alive or not. So we are currently in discussion with Corinne, that is the EMA leader of this special interest group, and we will then decide whether to keep it active. Thank you for the question. We will come back on that. Okay, Susanna, if there are no further questions.
I think
Thank you,
we
Christine.
may close the meeting. Thank you all the presenters and all the participants.
Thank you also to the colleagues from the Secretariat at EMA and from the audiovisual. Without this would not have happened. So thank you very much. And thanks to everyone. Goodbye.
Thank you. Have a good rest of the day. Bye-bye.
Bye.